We have addressed the urinary system, and we're going to look at the embryology of the urinary system, any malformations that can occur, the histology of the urinary system. That's what we are going to look at. First, I'd like us to start with the histology of the urinary system, and then after that, we'll go back to embryology, having known the various parts of the urinary system. That is how I'd like us to address this. Now, the urinary system, like we had discussed before, originates from the intermediate mesoderm, very close to the origin of the urogenital system, the genital system, so close to the gonadal ridge. When you're looking at the reproductive system, we said that there are two tubular structures that do arise. That is mesonephric tubule and also the Mullerian duct. Mesonephric tubule is also called Wolffian duct. And then we talked about the intermediate mesoderm. The cells in the intermediate mesoderm, once they have been initiated, they proliferate. And as a result of this proliferation, there is thickening of that intermediate mesoderm right from the cephalic region to the sacral region. So a thick part that, for the case of the urinary system, we refer to as the nephrotom, For the case of the reproductive system, we refer to it as the gonadal ridge. So both of them constitute what you call urogenital ridge. So the nephrotom for the urinary system and the gonadal ridge for the gonads. That is how the intermediate mesoderm is divided into those two areas. So I'd like us to first address the functions of the urinary system. And after addressing these functions of the urinary system, we'll go to see how is the structure of this urinary system suited to the function. How do they match to that function? One of the most important functions is removal of metabolic waste products. During metabolism, we have things like urea being produced, nitrogenous waste products being produced, and those must be removed from the body. Accumulation of this in the body can cause damage to the blood vessels. If you have uremia, this can cause damage to the blood vessels. Then you can end up with edema and subsequently kidney failure, because the blood vessels of the kidney are very, very narrow. Another one is regulation of body fluid. Regulation of body fluid in terms of reabsorption of water. So when you reabsorb water, you regulate body fluid. When you reabsorb water, then you are increasing water within the circulatory system. That way, you are conserving water, and also, as a result of increase of water in the circulatory system, you are increasing pressure, blood pressure. So to maintain blood pressure, you need the kidney to be able to reabsorb this fluid. Otherwise, if water is lost, then the body becomes dehydrated. So the kidney developed as a result of movement of the primitive animals from the aquatic environment, where they were surrounded by water, to a terrestrial environment, where they are not surrounded by water anymore, but we take the water through drinking water or through the food we eat. And therefore, as a result of this, we lose more water when you are exposed to the environment compared to the animal which is within the water. And therefore, that kidney had to develop to conserve water. As a result of this conservation, even the secretory product from the kidney is also important. For humans and mammals, usually you secrete fluid which is liquid, as a waste product of urea. For the birds, you have uric acid, which is solid. Otherwise, the bird will have to have a bladder, and that will be carrying a lot of weight. So regulation of body fluid is very important. The production of hormones, we've already looked at the endocrine functions of the kidney. And if you remember, we talked about erythropoietin. Erythropoietin, when we are talking about the formation of the blood cells. So erythropoietin is very, very important in the formation of the blood cells. Apart from that, we have renin, which is produced within the kidney by juxtaglomerular cells. That renin acts on angiotensinogen, converting it to angiotensin 1. The angiotensin 1 then is taken to the liver, where it is the lung, where it is converted to angiotensin 2. Angiotensin 2 is a vasoconstrictor. So angiotensin 2 can also increase blood pressure by virtue of constriction, vasoconstriction. But angiotensin 2 also is very important in acting on the cortex, spinal cortex, specifically the zona glomerulosa, to produce aldosterone. And the aldosterone now is transferred to the kidney, where you have reabsorption of sodium in the distal convoluted tubule. As a result of the reabsorption of sodium, you then have passive reabsorption of water. That increases the blood volume, and therefore the blood pressure goes up. So that is very important in terms of conservation of water, increase in body fluid, regulation of body fluid, and also regulation of blood pressure. So we need to be cognizant of that. We have others like calligreen, which is vasoactive agent, usually peptides produced by the cells in the proximal convoluted tubules, also in the distal convoluted tubule. The other function is secretion of autocoids. Autocoids are hormone-like peptides or secretions, hormone-like. They are some of them are amines, and these hormone-like secretions are secreted by the cells of the kidney, the proximal convoluted tubule, and even in the distal convoluted tubule. These autocoids or hormone-like structures have a paracrine or autocrine activity. That means the hormone is not released into blood circulation, but it is released to the environment of the cell and will have an effect on the cells immediately next to that releasing cell. Something like nitrous oxide is an autocoid, and these prostaglandins are autocoids, and these ones here influence the dilatation of the blood vessels or constriction of the blood vessels, and therefore have an effect on the glomerular filtration. And that is important for us to know. The other important thing is regulation of acid-base balance. During passage of urine, we usually have acidification of urine. That means hydrogen ions are excreted to the outside. When the hydrogen ions are excreted to the outside, one of the byproducts from that metabolic activity and secretion of hydrogen ions is the bicarbonate ion. So that is going to help in maintenance of the alkaline nature of the blood. So that is very important in regulation of acid-base balance. The other one is production, storage, and excretion of urine. Production of urine as an ultrafiltrate of plasma. Production of urine as an ultrafiltrate of plasma. About 25% of the cardiac output passes through the kidney, 25%. And in a day, what is filtered through, the plasma that is filtered through for a whole day, is about 180 liters. 180 liters of fluid is filtered through at the glomerulus. That is during the ultrafiltration. Out of that, over 99% is reabsorbed. So you have something like 1.2 liters of urine being produced per day, whereas 180 was actually filtered as primary urine. After that primary urine, it's then the water and the ions and also the glucose and all those are reabsorbed back into circulation so that you don't lose. Some of the proteins, amino acids are reabsorbed into circulation. So what is lost is the waste products, and what is lost. And that is important to note that the kidney performs a lot of function, very big function for the whole day. Imagine 108 liters passing through the kidney. And then all that, almost everything must be reabsorbed until you remain with only about 1.2 liters, which is now lost as urine. The other function is secretion. Secretion, not necessarily alcohols alone, but secretion of other products. Secretion of iron, secretion of creatinine by the cells. So that is important. So we need to look at these functions. Where is reabsorption taking place? Where is the ultrafiltration taking place? So that we understand that function of the urine in terms of the blood flow, in terms of ultrafiltration to form the primary urine, and then reabsorption and secretion so that you end up with the urine, secondary urine, which we now pass out as urine during micturition. Now, when you look at the kidney as an organ, the kidney is usually covered by a capsule, just like any other organ. It's covered by a capsule. It contains connective tissue. It also contains the particular cells that are functioning on the kidney. When you cut a fresh kidney, we know there will be a capsule on the outside covering it. This part of the kidney where the blood vessels enter and where the ureter enter, where the nerves enter, we refer to as the hilum of the kidney. The hilum of the kidney. Once the blood vessels enter, they divide until at this region, which is the corticomedullary junction, which they form aqueous vessels. Aqueous vessels. So this part of the kidney we refer to as the medulla. The medulla, the inner part is the medulla. And the medulla consists of mainly collecting ducts, and also the loops of Henle. Most of it are collecting ducts and the loops of Henle, part of the nephron. The outer part here, which is appears granular, is the cortex. So we have the cortex as the outer part next to the capsule, and then the medulla. You will notice that part of this medulla extends from the corticomedullary junction. This is the area we refer to as the corticomedullary junction, and open into this area. And in this area, we have two structures. One, each of these structure extending all the way from the corticomedullary junction and having around a tip. This tip we call the papilla. The papilla. So this part here is the papilla. That's a papilla. And this is where the opening into another larger duct that we refer to as the minor calces. Minor calces. And the structure that is broad at the base and very narrow at the apex we refer to as the pyramid of the kidney. That is the pyramid. So this structure is the pyramid, and its apex is the papilla, and at that apex is the opening of the minor calces. So each papilla opens into a minor calces where the urine from the collecting ducts now are emptied into the papilla, at the papilla to the minor calces. So that collecting duct at that region, we refer to it as the papillary duct, the papillary duct. When they empty into the minor calces, several minor calces collect together and form some major calces. So several of these empty into a major calcis, and those major calcis now opens into a renal pelvis. The renal pelvis forms the beginning of the ureter. The renal pelvis forms the beginning of the ureter. Around the kidney, we have a lot of adipose tissue, which is very important in protections of the kidney. It's cushioning to the kidney. So we are going to see how do these minor calces originate from, how do these papilla originate from, and how do these collecting ducts originate from, and what is the significance of this corticomedullary junction, where we have the arcuate vessels. From those arcuate vessels arise the afferent vessels that now supply the glomerulus, what you call the glomerulus. So it's very important to note that. So renal pelvis is the beginning of the ureter. And what is the structure? So remember, minor calcis is where the urine is emptied from the collecting ducts. So from that minor calcis all the way to the kidney, all the way to the urinary bladder, urine flows by peristaltic activity of the muscles from the minor calcis to the urinary bladder, where it is stored. That peristaltic activity is because within the minor calcis, we have some specialized muscle structures that act as pacemaker cells. They act as pacemaker cells. So they initiate that peristaltic activity. Just for gross anatomy, you'll know that the kidney is the posterior abdominal wall. It's covered by the ribs, the lower part. The right kidney is low due to the development of the liver, so that pushes it to the lower part. The left kidney is slightly higher compared to the right. It receives 25% of the cardiac output, which I've said, through renal artery. But occasionally, when we talk about development, we will see that we also can have some aberrant blood vessels apart from the main renal artery. And total blood plasma passes through the kidney in 15 minutes. Total blood plasma passes through in 15 minutes. So you can just imagine, without a kidney, how much of the waste products will be retained in blood. For the relations, now, you just need to know that it is close to the posterior abdominal wall, but in the lumbar region. It's in the lumbar region. So we're going to look at the ascent of the kidney from the pelvis to the lumbar region. Posteriorly, it's related to the diaphragm, the psoas muscle, the quadratus lumborum muscle. And then we have the nerves. The nerves, both from the intervertebral spaces, the subcostal nerves, iliohypogastric nerves, ilioinguinal nerves. This just to show the kidney, but notice that the relationship is different. The relationship is different. You have suprarenal gland in the upper part. That is the superior pole of the kidney. Whereas on the right, you have the duodenum. You have the liver, part of the liver. You have the colon, and you have the small intestines. Whereas on the left, you can see that we have, of course, the suprarenal gland is there. We have the pancreas instead of the duodenum. We have the intestines on the medial, and then we have the colon. So these are important relations of the kidney, so that any structure to which it is close to could easily cause some injury to the kidney, or infection can spread to the kidney. But remember, there's a capsule which can sequester the infection either within the capsule of the kidney or outside the other organs. For the blood supply, we talked about renal artery. Remember, 15 minutes. The renal artery supplies blood. Total plasma is within 15 minutes is already filtered through. In a day, 180 liters has been filtered through. So every time 25% of the cardiac output goes, it's going to pass through. And you can imagine 25% of cardiac output, you can multiply that so that you know in 15 minutes how many liters have been released. The renal artery will divide into a posterior and anterior branches. And then from there, we have interlobar branches, the arcuate at the corticomedullary junction. And from the arcuate, we have interlobular branches. And apart from this, we also have from the interlobular, one branch that bypass the cortex, to supply the capsule. So we refer to that as capsular arteries. Otherwise, in the interlobular branches, we now have the afferent arterioles. From these afferent arterioles, the blood goes to the glomerulus, which is the tuft of capillaries. And after the glomerulus, we have the efferent arteriole, and then the efferent arteriole now goes to form what we call vasa recta. So the vasa recta is a capillary network that now surrounds the loops of Henle. That's referred to as the vasa recta, dipping all the way to the medulla, in the medulla. So here we are talking about an arterial, a capillary network in the glomerulus, and another capillary network network interruption. So from the efferent arteriole, you have vasa recta, and then interlobular, the arcuate, interlobar, and then the renal vein. So that is how the blood flow to the kidney is concerned. Of importance for us that we need to be very careful in terms of urine formation is from the afferent arteriole, the glomerulus, and the efferent arteriole. This is important. And at this stage, I would like to say that the efferent arteriole has a smaller diameter than the afferent arteriole. Has a smaller diameter than afferent arteriole. So that we have the pressure. We can create some pressure by that. We increase glomerular filtration rate because you are increasing the pressure since a lot of blood has now to pass through a narrow vessel, and therefore there will be more fluid being lost in terms of ultrafiltration at the glomerulus. This is the structure of the glomerulus. This is the structure of, these are the blood vessels. We have injected some dye into some solid latex into the blood just to demonstrate that the capillary network that we refer to as the glomerulus is fed by a large vessel. This is the large vessel, the afferent arteriole. This is the efferent arteriole, which is a slightly narrower vessel. So pressure here is increased so that more fluid can now be filtered through into the lumen of the Bowman's corpuscle. We have two types of nephrons. The functional unit of the kidney is a nephron. The functional unit is a nephron because this structure that is called the nephron is repeated over and over. We have two types of nephrons. We have the cortical nephrons, which is mainly in the cortex of the kidney. And then we have juxtamedullary nephrons. Juxtamedullary nephrons start at the junction between the cortex and the medulla. And their long loop of Henle is very thin. They have a very thin, long loop of Henle, which is entirely in the medulla. Whereas in the cortical, they have a very short, thin limb of Henle. A very short, thin limb of Henle, which is not entirely in the medulla. So this has a better chance of exchange, what we refer to as countercurrent exchange or countercurrent multiplier exchange, which physiologists will be able to address to you. The other component that we have is the collecting ducts, and the collecting ducts are the arcuate collecting ducts, arcuate, forming something like an arch. These then go into the intermediate collecting ducts, and at the lower end, the papillary collecting ducts. The papillary collecting ducts. Usually, sometimes we have a collection of the collecting ducts extending all the way from the cortex to the medulla. Groups of collecting ducts extending from the cortex to the medulla. Those are referred to as the ducts of Bellini. Ducts of Bellini. These are just large collecting ducts which are aggregated together, extending from the cortex all the way to the medulla. Ducts of Bellini. We will therefore look at this nephron before we go to the collecting duct. And the nephron originates from the renal vesicle. We have a renal vesicle, and this renal vesicle is originating from the mesoderm, mesenchyme tissue. Mesenchyme tissue within the nephrotom. Mesenchyme tissue in the nephrotom. The condensation of that mesenchyme tissue will form initially a long vesicle, a fluid-filled cavity that is vesicle, and this vesicle elongates to form a sigmoid shape. A sigmoid shape. One part of the sigmoid shape becomes flattened, and around that flattened part, we have invagination of blood vessels from the aorta, abdominal aorta. This invagination of the blood vessels form a capillary network that we now refer to as the glomerulus. So the nephron originates from the renal vesicle. The nephron comprises of a renal corpuscle. This part we refer to as the renal corpuscle, a renal corpuscle. And this renal corpuscle comprises of a Bowman's capsule. A Bowman's capsule and the glomerulus. Bowman's capsule and the glomerulus form a renal corpuscle. The glomerulus is a tuft of capillaries. A tuft of capillaries between the afferent arteriole and the efferent arteriole. A tuft of capillaries between afferent arteriole and efferent arteriole. That's what we refer to as glomerulus, a capillary network. Then we have a Bowman's corpuscle. A Bowman's corpuscle comprises of two parts. An outer part that we refer to as the parietal part of a cup-shaped structure. The outer part is a parietal part, forming like something like an outer part of the cup. And this outer part is lined by simple squamous epithelia. Simple squamous epithelia. That simple squamous epithelia is what we refer to as epicyte. And those do not participate in urine formation. The visceral part of the Bowman's corpuscle contain some specialized cells that we call podocytes. This is a podocyte. This is a podocyte, And this specialized cell contain a lot of processes, some large processes that we refer to as the primary processes. And these primary processes give rise to smaller processes that we refer to as secondary processes. This we refer to as the podocytes. Podocytes, and between these podocytes, the secondary processes, we have some narrow spaces. These spaces, separated by the podocytes, form what we refer to as filtration slits. Filtration slits. And it is through these filtration slits that the fluid passes during ultrafiltration from the blood vessel into a Bowman space. The Bowman space. The Bowman space is the space between the visceral part and the parietal part of a Bowman's corpuscle. The processes of the podocytes surround the blood vessel. They surround the capillaries. They surround the capillaries. So when they surround these capillaries, then we have that filtration slits. That you need to note. Within this space of the where we have the podocytes, we also have other cells. Cells that we refer to as mesangial cells. Intraglomerular mesangial cells. Intraglomerular mesangial cells. What will be the functions of these intraglomerular mesangial cells? One of the main functions is secretion of the mesangial matrix. Secretion of the mesangial matrix. And the podocytes, the endothelial cells, the blood vessels are surrounded by this mesangial matrix. And the matrix is the area where we have exchange of or passage of material. So the matrix plays, the mesangial cells plays a very important role in terms of that secretion of the mesangial matrix. But because they are also interspersed between the mesangial cells, they act like pericytes, and the capillaries, they act like pericytes. They act like pericytes. They are also capable of contracting. So they have actin within the cytoplasm, so they can contract and therefore regulate the blood within the capillaries. That's very important. Apart from this, during this ultrafiltration, some of the substances might not be able to pass through and accumulate within the mesangial matrix. Some of the antibody-antigen complexes, for example, might not be able to pass through. The mesangial cells phagocytize. They phagocytize this material, so they help up in cleaning the filtration bed, ultrafiltration bed. Apart from that, they also secrete some outercoids. Outercoids, which are very important. So that is the function of the intraglomerular mesangial cells, which lie between the podocytes and also the endothelial cells. And you can see how by modifying, by causing modification of the capillary diameter, they can be able to regulate part of the flow of blood and therefore influence urine formation. That is very important. That is within the Bowman's corpuscle. Now, the Bowman's corpuscle has got two parts. The part where the blood vessel enters, the part where the blood vessels enter and leave, this is what we refer to as the vascular pole. The vascular pole. And then after ultrafiltration, the primary urine leaves from the vascular pole and enters the proximal convoluted tubule. So that area where they leave, the proximal convoluted tubule, now as primary urine, is what we refer to as the urinary pole. So we have a vascular pole and a urinary pole. So urine passes to the proximal convoluted tubule. Proximal convoluted tubule. What happens at the proximal convoluted tubule? Let's just look at the structure. This is the glomerulus. Well, what we have here, these are glomeruli, what is appearing as red. The cells in here are the podocytes and also nuclei for the mesangial cells and endothelial cells. So this is what we refer to as the Bowman's space. Bowman's space. This is the vascular pole of the Bowman's corpuscle or renal corpuscle, the vascular pole. So we have the arteries entering and leaving. Now, when they leave, they enter into the urinary pole, and the urinary pole comprises of cells like these ones here. They comprises of the proximal convoluted tubule, comprises of simple cuboidal epithelia. Simple cuboidal epithelia. The simple cuboidal epithelia have a large central place nucleus. The apical part of the simple cuboidal epithelia has a lot of microvilli. The microvilli is to increase surface area for reabsorption. Reabsorption. At the basal part, the membrane has a lot of basal infoldings. These basal infoldings form a basal labyrinth, also to increase surface area. Between these basal infoldings of the lower part of the membrane, we have a lot of mitochondria. A lot of mitochondria. So part of this basophilic staining is because of mitochondria. So we have an increase in the basal, increase in surface area. The mitochondria is to provide the energy. Increased surface area by the labyrinth. Mitochondria provides the energy because we have active transport of ions. So in the proximal convoluted tubule, water is reabsorbed, amino acids are reabsorbed, ions are reabsorbed, sodium, potassium, ions, all those are reabsorbed in the proximal convoluted tubule. So we need active transport, and that's why we have striations at the basal part, and mitochondria to provide that motive energy. And then reabsorption of water, reabsorption of amino acids, all that is because we have an increase in surface area by microvilli on their apical luminal surface. That is the structure of the proximal convoluted tubule. After the proximal convoluted tubule, we go to the loop of Henle. Like I've said, the loop of Henle comprises of two parts. It comprises of the thick end and a thin end. Thick end and a thin end. And I've said the juxtamedullary nephron has a very long, thin end of the loop of Henle that is located entirely in the medulla. Entirely in the medulla. The thick parts are similar to the proximal convoluted tubule. The thin ends are lined by simple squamous epithelia. Simple squamous epithelia. That lines the thin ends of the loop of Henle. It also comprises of two parts, the descending loop of Henle and an ascending part of the loop of Henle. The descending loop of Henle is permeable to water. So water can be reabsorbed back into circulation. Between these loops of Henle, we have the capillary network that we refer to as the vasa recta. Vasa recta. So water can still be absorbed into circulation, into blood, because the proximal convoluted tubule is permeable to water. The distal convoluted tubule is not permeable to water. It's impermeable to water. It's not permeable to water. But ions can pass through, can be reabsorbed into the distal convoluted tubule, the ascending part of the loop of Henle. Because of this, you have an osmotic gradient, concentration gradient, a concentration gradient within the medulla. And this is where we have the countercurrent principle of exchange, what we call countercurrent multiplier effect. From the loop of Henle, the thin ends of the loop of Henle, we have the thick end of the loop of Henle, which is the same as the distal convoluted tubule. What is the structure of the distal convoluted tubule? In this slide, this is the distal convoluted tubule. You can see the staining is light staining because there's less mitochondria. It's also lined by simple cuboidal epithelia, and these simple cuboidal epithelia have very few microvilli at their luminal surface, unlike what you have in the case of the proximal convoluted tubule. And therefore, it appears as less basophilic because of less mitochondria. In the distal convoluted tubule, we have further reabsorption of ions, and also to some extent reabsorption of sodium. That reabsorption of sodium is under the influence of aldosterone. The aldosterone causes reabsorption in the distal convoluted tubule, and that aldosterone, by virtue of reabsorption of sodium, you also have reabsorption of water. So water gets back into the blood from the lumen, and therefore we are helping concentrate urine. We are helping in concentrating urine. So this way we have further reabsorption. At the distal convoluted tubule, we also have excretion of hydrogen ions. So hydrogen ions are excreted by the cell into the lumen. So this is where we have acidification of urine. Acidification of urine. Now, there is a very specialized part of the distal convoluted tubule which lies close to the afferent arteriole. The part which lies close to the afferent arteriole of the distal convoluted tubule, the cells are very closely packed together and appears columnar. Very closely packed together and appears columnar. Only at the part close to the afferent arteriole. The part away from the efferent arteriole, the cells are far apart, just like the other part of the distal convoluted tubule. These cells are referred to as the macula densa cells. Macula densa cells is M-U-C-L-A. Densa, D-E-N-S-A. The macula densa cells are these specialized epithelial cells within the distal convoluted tubule close to the afferent arteriole. They normally act as osmoreceptors. They sense the concentration of sodium in the distal convoluted tubule, the lumen of the distal convoluted tubule. So after sensing the concentration of sodium chloride, then it can give feedback also, one, to the suprarenal cortex, the zona glomerulosa, so that you can have either more aldosterone being produced or less aldosterone being produced. And that, of course, will also affect the glomerular filtration rate. So this is a sort of feedback mechanism, that a high concentration within the lumen will be detected, and this can give now a feedback by release by those cells of adenosine, and that adenosine causes afferent arteriole to vasoconstrict. So if the afferent arteriole vasoconstricts, then you have less ultrafiltration. And that is very important for urine formation. It's also very important in that when you have low sodium chloride in the urine, sodium specifically, then that also can affect the renin production by juxtaglomerular cells, so that you can have the renin angiotensin aldosterone pathway. So that is very important. The other cells which lie very close to the vascular pole, the blood vessels, the afferent arteriole, the smooth muscle in the tunica media. The smooth muscle in the tunica media at the afferent arteriole comprises of specialized cells. These specialized cells are referred to as juxtaglomerular cells. Those juxtaglomerular cells are the ones that produce renin. They secrete renin. So you can see the relationship between macula densa cells and juxtaglomerular cells in terms of production of renin. Apart from this, we also have some flattened cells. These flat cells, again, close to this junction, close to the macula densa cells, and also the wall of the afferent arteriole. Close to the wall of the afferent arteriole, where we have juxtaglomerular cells. These are referred to as extraglomerular mesangial cells. Extraglomerular mesangial cells, also called Lassis cells. LACIS, or also called Pulkisen cells. Lassis cells or Pulkisen cells. What is the function of these cells? They are flattened cells. They lie between juxtaglomerular cells and the juxtaglomerular cells and also the macula densa cells. What would be their function? Just like the intraglomerular mesangial cells, they provide structural support to the juxtaglomerular cells. And because they lie in that area, these cells constitute what you call juxtaglomerular apparatus. So the extraglomerular mesangial cells or Lassis play a role in supporting the juxtaglomerular apparatus. But they make contact with the macula densa cells, make contact with juxtaglomerular cells. So they are very important in cell-to-cell signaling. And that is very important for the secretion of renin. As the macula densa cells detect the sodium concentration, then this is then detected also by the extraglomerular mesangial cells. They also secrete some autocoids. Autocoids, these are like prostaglandins, are autocoids. which can influence prostaglandins or nitrous oxide, can influence also the smooth muscles within the wall of the blood vessels, can cause vasoconstriction or vasodilatation. And just like the other one, they also help in phagocytosis. In phagocytosis of any dead cells within that area, this can be phagocytized by the juxtaglomerular, extraglomerular mesangial cells. So that is very important, and we need to note that these cells, comprising of macula densa cells, juxtaglomerular cells in the afferent arteriole, extraglomerular mesangial cells, these constitute what you refer to as juxtaglomerular apparatus. Juxtaglomerular apparatus. And you can see how this can influence urine formation. From the distal convoluted tubule, if you notice what we're looking at in this diagram, the distal convoluted tubule, we then have the collecting duct. This empty into the collecting duct. But at the distal convoluted tubule, we also have reabsorption of water. So the urine is further being concentrated, reabsorption of water, during that passive absorption of sodium by aldosterone. We have the collecting ducts starting from the arcuate, the most cortical parts, the intermediate, and the papillary. So the arcuate will be lined by simple cuboidal epithelia, and as you go down to the papillary, those ones are columnar epithelia, where they open into the papilla, Now notice the way they enter into the common duct here, and we'll be discussing that when you're talking about development. Now the collecting ducts originate from the ureteric bud. Embryologically, we have something that's called ureteric bud, and all the collecting ducts originate from the ureteric bud. The collecting ducts also are sensitive to ADH, vasopressin hormone, which is produced by the posterior part of the pituitary glands. Remember, it is actually synthesized in the hypothalamus by supraoptic and paraventricular nucleus, transported to the posterior part of the neurohypophysis, where they are stored and released. So when they are released, they act on the collecting ducts, because those collecting ducts have aquaporin channels. And there we have further reabsorption of water. That is why from 180 degrees and 180 liters, only 1.2 liters are eventually released into the bladder, which is now passed out as urine. So the papillary part of the collecting ducts empty into the renal papilla, into the minor calces. So here we have the glomerulus, the Bowman's corpuscle, both the epicytes, which are flat here, and the podocytes, epicytes and podocytes. And these are the vasa recta. So this is a global view of the kidney, looking at the cortex, and then looking at the medulla. And we said earlier that some ducts extend all the way from the cortex. They extend all the way from the cortex to the medulla collectively. This we refer to as the ducts of Bellini. This is the medulla. In this slide here, we have a capsule made up of fibrous connective tissue, some bit of adipose tissue. This is the capsule, and that's covered by adipose tissue for protection. And these are the ducts. These are the proximal convoluted tubule and some of the distal convoluted tubule close to the cortical part. This is, again, a Bowman's corpuscle, what we call renal corpuscle. The renal corpuscle comprising of the blood vessels here that form capillary network that is referred to as the glomerulus, and then the Bowman's corpuscle. The Bowman's corpuscle that comprises of the podocytes, the visceral part, the podocytes, the mesangial cells, and then a Bowman's space where the urine, primary urine, is released into, and the parietal part of the Bowman's corpuscle comprising of flat cells we refer to as the epicytes. Epicytes. And then at the distal convoluted tubule, we have the macula densa cells, part of the distal convoluted tubule, and then the juxtaglomerular cells, which are part of the wall, specialized muscle cells within the wall of the afferent arteriole. And then the other cells that we refer to as extraglomerular mesangial cells. So all these cells here are suspended within the glomerular matrix, within the Bowman's corpuscle matrix, what we call mesangial matrix, that is secreted by the intraglomerular mesangial cells. Notice distal convoluted tubules and the proximal convoluted tubule very deeply staining basophilic basal parts because of the mitochondria. We have a microvilli on the surface as compared to this where the luminal part looks smooth. This one, the luminal part is slightly rough because of a lot of mitochondria. From the collecting ducts empty into the minor calces, which empties into the major calces, which empties into the ureter. From the minor calces, the structure will be the same up to the ureter. We have an epithelia that line the lumen that we refer to as the transitional epithelia. So we have a transitional epithelia lining the lumen. So it lines the entire urinary tract. Sometimes it's called uroepithelia. Uroepithelia. Resting on a basement membrane. Then we have a lamina propria. Lamina propria consisting of loose connective tissue. And after the lamina propria, we have a muscular layer. The muscular layer comprising of inner longitudinal, middle circular, and outer longitudinal. Although they say longitudinal, the muscles appear spiral in nature. They spiral around. That is from the minor calces, also in the major calces. But like I said, some smooth muscle cells in the minor calces form what you call pacemaker cells. Then from the minor calces to the major calces is the same structure. And then the major calces, several major calces empty into the renal pelvis. The word renal pelvis is a basin, comes from the word basin. So the renal pelvis appear like a basin in shape. So it collects all that urine. A basin normally is for collecting fluid. So it collects the urine, and then the urine passes to the ureter. This is the ureter. You look at the lumen, it is stellate in shape, and this is because the muscles have contracted. So it is stellate, and we have the epithelia here is transitional epithelia. Transitional epithelia. Why transitional? Because transitional epithelia is capable of stretching or being stretched. When it is completely empty, this is how it would appear. When it is full of urine, then the lumen becomes more or less rounded. The cells become flat. The cells become flat. And that is important. We have dealt with that when we were looking at, remember when we were looking at the epithelia, we talked about the shape of the cells as being dome-shaped. Dome-shaped. When they are dome-shaped, usually when it is empty. When it is full, they become flattened. And that's why we refer to it as transitional. The ureter extends all the way from the renal pelvis up to and enters into the urinary bladder at an angle. We have a lamina propria comprising of loose connective tissue, what appears as blue here, green. This lamina propria with the loose connective tissue. Then we have the muscular layer here, and the muscular layer will be comprising of inner longitudinal, when you see some fibers here, they have been cut longitudinally, a middle circular layer, and an outer longitudinal. But like I've said, it appears spiral. And that's why you don't see a very nice circular arrangement. So it's inner longitudinal, middle circular, and outer longitudinal. And it is this muscle that has peristaltic activity contractions transporting urine from the urinary bladder, from the kidney, from the renal pelvis, up to the urinary bladder. And like I said, it enters the urinary bladder at an angle. After that, we have the outer part here, which is the adventitia, and the adventitia contains some connective tissue, dense connective tissue, some blood vessels, like the artery here, some nerves, all that is within the adventitia. Now, almost each of these nerves almost each of the muscles has a nerve supply. So that when you have a kidney stone, usually it will be very, very painful, very painful. So that's what we have as far as the ureter is concerned. Urine passageway is not a continuous flow. Urine actually enters the urinary bladder in jerks. So it's parted into the urinary bladder because of that constriction. Because of contraction, peristaltic contraction of the urinary bladder. Peristaltic contraction within the urinary bladder, but peristaltic contraction of the ureter, which extend all the way to the urinary bladder. So that is what we have as the structure of the ureter. Now, the ureter enters the urinary bladder at an angle. It enters the urinary bladder at an angle. Because of that, there is a very thin, part which covers the entry of the urinary bladder on the luminal side. So when the urine fills up, it pushes this membrane outwards and therefore shuts the entry into the bladder. This is what we refer to as shutter valve mechanism, a shutter valve mechanism. And this shutter valve mechanism, therefore, ensures that urine does not flow back into the ureter. Urine does not flow back into the ureter. So we do not have what is typically the anatomical sphincter. We do not have an anatomical sphincter at the entry of the urinary bladder into at the entry of the ureter into the urinary bladder. That also is a potential area of constriction. A potential area of constriction where a kidney stone could very easily lodge. The other area where a kidney stone would lodge is at the beginning of the ureter. From the renal pelvis, we said the ureter starts from the renal pelvis. So at that area, that is a potential area of constriction. And also, as it passes into the pelvic cavity, at the pelvic brim, as it crosses the iliac vessels, the ureter can also be constricted in that particular area. So those are the three potential areas of constriction where you can have a kidney stone lodging. So urine goes to the urinary bladder, and the structure of the urinary bladder is also adapted. It's adapted to be able to store the urine and to be able to extrude the urine in the process of micturition. The process of micturition. So what do we have as the histology of the urinary bladder? First, we have the fundus of the urinary bladder. So we have a fundus, which is the area above the entry of the ureter. At the position where the ureter enters and at the opening of the urethra, we have a triangular area we call the trigone. The trigone. The trigone is of endodermal origin. It's locally thought to be part of the allantois. That's the triangular area. So the urinary bladder has an epithelia, and that epithelia comprises of transitional epithelia. Transitional epithelia. The transitional epithelia, remember, is an epithelia that's capable of stretching, capable of being stretched. So when the bladder is full, it becomes almost flat. When the bladder is empty, the cells at the outermost layer are dome-shaped. Dome-shaped. Then we have a lamina propria, like all the other lamina propria, comprising of loose connective tissue with a lot of elastic fibers. Now, why do we have fibers? Because these elastic fibers are very important when the bladder is stretched. The elastic fibers are stretched, and during the emptying process, the elastic tissue also contracts. And then we have a submucosa. And this submucosa contains vessels. You can see some vessels here. So this lamina propria, epithelia, lamina propria, submucosa with blood vessels here, larger blood vessels, some lymphatics. And then after that is the muscular layer. So epithelia, lamina propria, submucosa, and then the muscular layer. This muscle is referred to as the detrusor muscle. Detrusor muscle, which is activated by the parasympathetic nervous system from S2, S3, S4. So during micturition, parasympathetic nervous system is important in extruding the urine because it acts on this smooth muscle that we refer to as the detrusor muscle. When you look at the muscle arrangement, we have similar to what we have in the ureter, an inner longitudinal, middle circular, and an outer longitudinal. But these three layers are not very clearly visible, not very clearly oriented. Not very clearly oriented because the cells might appear as spiral. So because of that, the muscles contract as a unit during expulsion of the urine. And then the outermost layer of the urinary bladder will comprise of tunica adventitia, where it is not covered by peritoneum, that is the anterior part of the bladder, and the lateral part of the bladder. It's not covered by peritoneum, so we refer to it as the adventitia, which is made up of connective tissue. And then the part which is covered by peritoneum, which is the superior part, and also the posterior part of the urinary bladder, will be covered by a serosa. Where we have a serosa, we'll have a subserosal layer having some connective tissue. So that is the structure of the urinary bladder. From the urinary bladder, urine is then released by the process of micturition, and we'll be looking at pontine micturition reflex center later on. Pontine micturition reflex center, which is very important in regulating that reflex activity where you now feel like you must go and pass urine. From the urinary bladder, urine then passes to the urethra. But we have two sphincters. We have the circular muscle of the urinary bladder becomes very thick at the beginning of the urethra to form an internal sphincter. Internal sphincter. And then part of that internal sphincter, now on the outer part, immediately after that, we have part of the pelvic diaphragm muscles, urogenital diaphragm muscles, forming an external sphincter. That external sphincter is formed by skeletal muscle. Skeletal muscle. So that is under voluntary control. The internal sphincter is under involuntary control. The external sphincter is under skeletal muscle. So from there you have urine passing from the bladder through into the, in the case of the females, into membranous part of the urethra, and then just a part of the, in the lower part of the clitoris, a small part which is almost homologous to the penile part of the urethra. So the urine then passes out. In the case of the males, at the neck of the urinary bladder, we have usually, we have the prostate gland. So the urethra traverses the prostate gland, and that's what we talked about in the morning, as being an area where you have potential constriction of the urethra when the prostate gland enlarges. At the area of the prostate gland, the epithelia is uroepithelia, transitional epithelia. Immediately after transitional epithelia, we have the lamina propria, and then we have the glandular tissue of the prostate gland, the fibromuscular tissue. In the case of the membranous part, similarly, it is that is where we have simple, sometimes pseudostratified to simple columnar, and towards the end, stratified by stratified columnar epithelia. And I think that's what we talked about last time in our practicals when we were looking at the reproductive system, where in the penile urethra, you have bistratified columnar epithelia extending up to the navicular, where you then have a dilated part of the urethra as it opens at the glans penis, where the epithelia also changes to stratified non-keratinized epithelia. Stratified non-keratinized epithelia. In the case of the females, the urethra opens at the vestibule of the vagina. vestibule of the vagina, which is surrounded by the labia minora, between labia minora and labia majora. That's what we have as the vestibule. That is the passage of urine formation and then passage of urine all through. Let's now look at how these organs develop. How do these organs develop? We have looked at the nephron as the functional unit. Nephron as the functional unit. And then we have nephron opening into the collecting duct. The collecting duct together with the nephron are referred to as uriniferous tubule. Uriniferous tubule. So the collecting ducts are not part of the nephron. They are referred as uriniferous tubule. And we have already said that the nephron originates from the renal vesicle. The uriniferous tubules originate from the ureteric bud. Ureteric bud. So let's now look at these structures. Where do they develop from? And how do they develop? We have already dealt with one structure. We have already dealt with the development. We have already dealt with the development of the male reproductive system and female reproductive system. And we talked of on the lateral side of the intermediate mesoderm. Remember, the paraxial mesoderm develops from the vertebral column. Vertebral column. The sclerotum forms the vertebral column. Lateral to that paraxial mesoderm, we have the intermediate mesoderm on either side of the vertebral column. So that paraxial intermediate mesoderm extends from the cervical region all the way to the sacral region. Cervical to the sacral region. But do these structures all develop to form the kidney? They all develop from the urinary system. During development, from the fourth week, usually from the fourth week of the embryo, after fertilization, we already have the intermediate mesoderm formed. And in the cervical region, we have condensation of the mesenchyme tissue that we refer to as the nephrotom. Condensation of the mesenchyme tissue extending all the way to the sacral region that we refer to as the nephrotom. Within the cervical region, we have cellular condensations. Cellular condensations that we refer to as the pronephros. So that nephrotom can be divided into three parts. We have the pronephros extending from the cervical region up to the thoracolumbar region, just close to the lumbar region, thoracolumbar region. And then at the thoracolumbar region and the entire lumbar region, we have the mesonephros. We call that the mesonephros. And then within the sacral region, we have the metanephros. So the nephrotom will be divided into those three segments. Within the pronephros, we have structures that form similar to the kidneys. We have some vesicle structures by condensation of the mesenchyme tissue. Condensation of the mesenchyme tissue, these form some primitive kidney that is not functional in man. It's not functional in man. So that disappears by the fifth week, it's already disappearing. In the fifth week, within the mesonephros, that is now in the area of the thoracolumbar region, we have development, the condensation of mesenchyme tissue forming tubular structures. On either side of the vertebral column. These tubular structures project towards the aorta, close to the aorta, projecting towards the aorta. And these tubular structures, several of them, at the distal part, they dilate to form a cup-shaped structure. This cup-shaped structure is invaded by blood vessels, forming a capillary network close to the cup-shaped structure. That we refer to as the external glomerulus. External glomerulus. Because it's not completely enclosed like what we have in a definitive kidney. Again, that is from the 5th, sometimes up to the 8th week, but this disappear. Again, urine is being formed at that stage. These tubular structures extending from the pronephros now collect into a common duct within the mesonephros that we refer to as mesonephric duct. If you remember when we were dealing with the male reproductive system, we had a mesonephric duct. And these structures are mesonephric tubules. So to some extent, they form some urine. They form urine because of the glomerulus. There is filtration, there's collection by these tubules which empty into the mesonephric duct. So urine formation is there. By the 8th week, by the 9th week, urine is being formed. From the 8th week, we have now extending caudally. There's an overlap during this development. As the pronephros disappears, mesonephros appear. As mesonephros disappear, the metanephros appear. By the 9th week, we already have urine formation. By the 9th week, we have urine formation. Now, the urine is formed, is transported by the mesonephric duct. Like we said earlier, it empties into the cloaca. In the caudal part of the mesonephric duct, in what we now refer to the area of the metanephric part of the nephrotom, we have a diverticula of a bud from the mesonephric duct in the sacral region. That posterior diverticula, a single diverticula on either side, extend towards the condensed mesenchyme cells, condensed or induced mesenchyme cells within the nephrotom. When they enter into the nephrotom, they induce those mesenchyme cells to form some condensation. That means they aggregate together and multiply. So the distal part of this extension now stimulate the mesenchyme cells to aggregate together and multiply. So they induce the mesenchyme cells to multiply. At the same time, those mesenchyme cells produce chemical substances which induce this diverticula to divide. So that diverticula from the sacral part of the mesonephric duct we refer to as the ureteric bud. Ureteric bud. Once it enters into the nephrotom, metanephric nephrotom, it divides into two and induce the mesenchyme cells now to continue multiplying. And those mesenchyme cells again produce chemical substances that induce the ureteric bud to continue dividing. That we refer to as reciprocating type of induction. Mesenchymal epithelial induction. The epithelia of the ureteric bud induce the mesenchyme tissue, that's mesenchymal induction, but the mesenchyme tissue also induce that epithelia so that you have division of the ureteric bud into two. That is what we are referring to as reciprocal induction. And this is what we are talking about here. That mesenchyme tissue condensation now forms what you call the metanephric blastema. Metanephric blastema, also referred to as the cape mesenchyme. Mesonephric blastema. Again, the mesenchyme tissue every time stimulate this to divide, to bifurcate. So there's continuous dichotomous division. Dichotomous division at every time induced by the mesenchyme cells. The mesenchyme cells induce again more proliferation. So as you can see here, there's an increase in size of the mesenchyme tissue within the blastema, but there is also division of the ureteric bud. The proximal part of the ureteric bud will form the ureter. The first 4 generations of divisions of the ureteric bud, the first 4 generations, coalesce together to form the renal pelvis. So we have a renal pelvis when we talk about the kidney, if you remember. We have a renal pelvis. And then the next generations, again, combine together to form a major calcis and then a minor calcis. So as it continues to divide, it forms a major calcis and a minor calcis. But also, note, this continue on for several generations, sometimes up to 17 generations. This is what we refer to as branching morphogenesis. Branching morphogenesis. Ultimately, at the most distal, after 17 generations, at the most distal part, the branch of the ureteric bud gives rise to a dilated part, the ambulary part, and the non-dilated part. It forms a dilated part and a non-dilated part. The non-dilated part now attaches to the renal vesicle that forms the nephron. The dilated part divides again into 2, a dilated and a non-dilated part. And that is why these structures normally are in the cortex. The corpuscles are in the cortex. The renal vesicle is in the cortex. So that's what we have as continuation. So all the time you have a dilated part and an undilated part. After 17th generation, that's what you have. Otherwise, the rest, you don't have that within the first few generations. So where they now completely at the end, the terminal parts of the ureteric bud form 2 non-dilated parts. The ambula, what they call the ambula is really dilated bud. The 2 non-dilated parts join to the renal vesicle, and that is where we have the arcuate type of collecting duct. And that's why we said from the minor calcis, the major calcis, the renal pelvis, the collecting ducts, the ureter, all those are from the ureteric bud, which is from the lower part of the mesonephric duct. Lower part of the mesonephric duct. And this is what we're illustrating here in the sacral region. So, remember, the ureteric bud is in the posterior part. It's pointing towards the posterior part. And then when it comes to the mesenchyme tissue, the metanephric mesenchyme at the posterior part, it's pointing posterior. It's in the pelvic region. So the kidney develops in the pelvic region. However, the last part of that development of the kidney is where we have the ascent of the kidney. Ascent of the kidney. The ascent of the kidney, eventually the kidney, like what we described before, is in the lumbar region. In the lumbar region, covered by the ribs, lying on the diaphragm anteriorly. So it's in the lumbar region. So it has moved from the pelvic bud to the lumbar region. The movement is not a physical movement. It's not a physical movement. What we have is differential growth of the vertebral column. So if you have a kidney fixed somewhere, the vertebral column continues to grow longer. As it grows longer, the kidney then appears to be in the lumbar region because the sacral segment has grown longer. Our lumbar vertebrae have grown longer. So that is what we have as the formation of the kidney. What can go wrong? Suppose now during that ascent, there is also a rotation. Remember, the ureteric bud points posteriorly, and the ureteric bud eventually forms the renal pelvis and the ureter, and those ones are medial. So there is a 90-degree rotation so that the hilum of the kidney now lies medially. What can go wrong? Suppose you do not have any ureteric bud. Absence of ureteric bud means there will be no induction of the metanephric mesenchyme, so there will be no kidney formed. That is a genesis of the kidney. A genesis of the kidney. The ureteric bud bifurcates into two only when it has entered into the metanephric part of the intermediate mesoderm. Only if it has entered in the metanephric part. So if it bifurcates prematurely, each of those eventually will form two separate ureters. So we can have a double ureter as a malformation. Each of these ureteric bud, as it divides, induce the mesenchyme tissue to form the blastema, which eventually coalesce together, fume together to form lobes, something that looks like lobes. In man, we do not have any lobes. In some animals, they are completely lobulated. So in man, all these coalesce together so that you have a very smooth outer margin, no particular lobes. In the earlier part, they might look lobulated because each of these induce growth of the mesenchyme tissue. So that is the development of the kidney. This is an early development of the kidney. Notice from the renal pelvis. Notice the duct systems as they go towards the cortical part, and notice the formation of the Bowman's corpuscle in the cortical part of the kidney, not from the hilum. Well, these are the structures we had looked at before: the ureter, the urinary bladder, and then the penis, the corpus spongiosum penis. And those who are there in the morning at the practical, you can notice that the corpus spongiosum penis is surrounded by a very thin tunica albuginea. And this thin tunica albuginea, therefore, ensures that during erection, although blood enters into the cavernous bodies here, during erection, the urethra is not completely closed up. Notice here the renal our renal pelvis, and the ducts. As yet, we do not have any glomerular formation here. And notice here, this is our distal convoluted tubule. This eventually developed to form Remember, look at these cells which are very closely packed together, very closely packed together. And then these ones which are very far apart. So these are the macula densa cells. So macula densa cells are formed at a very early stage. Sorry, not macula densa cells. This one here is the Bowman's corpuscle being formed. Bowman's corpuscle, this is the epicite, and this is the Bowman space, and these are the podocytes. Podocytes. So these are renal vesicles. So agenesis is one where we have said there is no ureteric bud, or poor development, that's hypoplasia, poor development because of problems of branching morphogenesis. Fewer branches means small kidney. Double ureter, we have talked about it, precocious division of the ureteric bud. Nephroblastoma, that's a cancer tissue. Polycystic kidney. Remember, we talked about this mesenchyme tissue, the intermediate mesoderm, in that nephrotome area, forming what you call a vesicle, a renal vesicle. That renal vesicle becomes S-shaped structure. So when this one part eventually flattens up to form a glomerulus, form a cup-shaped structure in which we have blood vessels entering, so that forms a Bowman's corpuscle. The other part attaches to the distal part of the ureteric bud. If you have a renal vesicle which does not attach to the ureteric bud, then fluid will collect within that vesicle because it contains a glomerulus. Fluid will collect into that vesicle, and then you have a lot of fluid-filled cavities. That's what we are calling polycystic kidney. Sometimes it is autosomal or recessive polycystic kidney. Sometimes the kidney fails to ascend to the lumbar region. It might be in the pelvic region or at the brim of the pelvis. During this development, part of the nephrotome on the inferior pole, they might fuse together. So when they fuse together, they form what is called horseshoe kidney, horseshoe type of kidney. And therefore, as it tries to ascend into the lumbar region, its ascent will be arrested by the inferior mesenteric artery. By the inferior mesenteric artery. And therefore, the kidney could be pelvic. So we call that ectopic. Ectopic. Sometimes we have supernumerary arteries. As the kidney ascends, it acquires new blood vessels. It acquires new blood vessels, and the old blood vessels disappear. Old blood vessels right from the pelvic region or sacral region disappear. However, some of them might persist, and therefore we end up with supernumerary extra arteries. The bladder, like we said, part of the bladder originates from the allantois. That trigone, they say, originates from the allantois. The allantois connects to the placenta. The allantoic duct connects to the placenta at the navel region. So because of this connection, the placenta at the navel region, sometimes that might persist. So that there is an opening connection between the navel region, an opening in the navel region up to the urinary bladder. That we refer to as urachal fistula, a urachal fistula. Because an allantois, when it's degenerated, it becomes what is called urachus. However, some parts of that urachus might fuse together, leaving some empty spaces. And these are cysts or sinuses. So if this persists, if a fistula persists, then a child will be born and urine will be dripping from the umbilical region because of the patent urachus, what we call patent urachus. Ectopic ureter That's what, when you have a supernumerary ureter, extra ureter, double ureter, the other one would not necessarily open into the bladder. It might open into the vagina, into the labia. So that becomes ectopic ureter. A vesicoureteral reflux. From the urinary bladder, urine can go back to the ureter. That one there is more of a nerve derangement, nerve tissue derangement. Remember, because between the bladder and the ureter, urine flows from the ureter. Urine flows from the ureter by peristaltic activity. So if there is weakening of the muscles of the ureter, so that peristaltic activity is not very strong, then urine, when it fills up, can pass back into the ureter. That's what you're calling vesicoureteral reflex. A congenital neurogenic bladder is where you have a bladder where urine is passing. There's a child is born. Usually, remember, the bladder has detrusor muscle, and the detrusor muscle, that's what controls, and also the nerve tissue from the sacral part controlling the external sphincter. So you might have an autonomous bladder where urine will just be flowing. Bladder extrophy, this is where you have failure of the anterior abdominal wall to completely close up. So the bladder might be exposed to the outside. So the bladder, the inner part of the bladder will be exposed to outside, and that is what we refer to as extrophy. So these are the malformations. And then for the urethra, we have at the labioscrotal swelling, the endoderm forms a urethral fold, a urethral meatus, and then urethral fold. This urethral fold connects together so that you have a complete closure and form the urethra. Sometimes there might be an opening of that urethral fold. And because of that urethral fold not closing completely, there will be an opening either on the lower side or the upper side of the penis. So we have what is called hypospadia. Hypospadia, if the opening is on the lower side, and epispadia, if it is on the upper side. So there could be a hole on the upper side when you urinate. Urine comes from the dorsum of the penis or on the ventral side of the penis, not at the glans penis. So that is where you have urethral problems, meatus not closing. But sometimes we have a valve at the opening of the urethra. That valve might persist. So if it persists, urine fills the bladder, but the urine cannot pass through because a valve is closing, a membrane-like structure closes the entry into the urethra. So a child will have a full bladder extending all the way to the abdominal cavity, and of course feeling a lot of pain, not passing urine, but when you pass a catheter, urine comes out because you have pushed the valve aside. This is to show double ureter. One ureter might open into the urinary bladder, the other ureter might open into the vagina. This is to show a horseshoe type of kidney where the inferior poles are fused together. The inferior poles are fused together, and its ascent is arrested by the inferior mesenteric artery. So it lies in the pelvic region. This is to show a polycystic kidney. You can see very large cyst, multicystic, a lot of cysts. And this is because of failure of the renal vesicle to attach to the distal part of the ureteric bud. This is to show aberrant blood vessels. You can see we are supposed to have only a single renal artery, but here we have several arteries supplying the kidney. This is a horseshoe kidney. horseshoe kidney, fused lower poles. The inferior poles are fused together because the nephrotom came very close together. And these are the mulcystic kidneys. And in cross sections, you can see dilated fluid filled spaces within the kidney. Fine, I'll invite any questions. If you have any questions. Any questions?